now I'm introducing Siân Culley. Siân Culley is a Royal Society University Research Fellow at the King's College in London. And she's interested in applying image analysis techniques to fluorescence microscopy. Is developing methods for microscopists in order to measure the quality of their acquired images and assess the biological information that can be accurately extracted from this data. Our talk will be about error and quality metrics in super-resolution microscopy and beyond. Thank you very much for accepting our invitation.
Thank you so much for inviting me. So yeah, this has been a super interesting workshop so far. And if there's one thing that I'm learning rapidly is that I thought I was a bioimage analyst, but I think I'm not. I think you guys are very much bioimage analysts, and I'm just a very standard microscopist. I run a research group where, like Rocco said, we're interested in methods development, but particularly interested in methods development to make life easier for users at the microscope and for bioimage analysts who have to deal with their data. So I had this talk all kind of ready to go, and then I realized that, again, when I normally do these kind of talks, I'm pitching towards either people I'm trying to get to use my software, or that I think should know about it, or developers who I want to show off to. And you are neither. So why should you care about anything I'm about to say, which I think is always a great place to start? So I'm going to talk about a couple of methods in this talk. One of them is single molecule localization microscopy. And are people using this? So this is just from a little PubMed search I did with a few keywords for super resolution, single molecule super resolution microscopy. And as you can see, in the last 10 years or so, there's been a big explosion in people using single molecule localization techniques. They're very popular, and there's a lot of image analysis associated with them. Right towards the end of my talk, I'm going to talk about some deep learning-based methods. Now, if we do that same search for deep learning and fluorescence microscopy, we can see uptake isn't huge, right? People aren't really using these techniques much outside of development. So those are the methods that people are using. And those are, so in your facility, you might, or whoever you work with, you might have quite a few people with this localization microscopy data. You'll have some people that might want to dabble with deep learning data. But also we need to look to the future, right? And so what are the methods that people are going to be using? So same search again, but here just in Nature Methods, where we publish all our new techniques. So again, in the last 10 years, there's been a kind of steady stream of a few developments in single molecule localization microscopy. It's still being developed. But deep learning, people are really developing these methods for microscopy. So even though people aren't using them right now, people are probably going to be using them soon, okay? So I'm going to start off by talking about single molecule localization microscopy and how we look at precision and error in these techniques. And this is work that I started in my postdoc with Ricardo Enriquez, and I'm still carrying on in my group at the moment. So just as a brief refresher for anyone who's kind of unfamiliar or has forgotten how this technique works, if we take a single wide field or confocal fluorescence microscopy image, this is a simulation, spoiler alert. I haven't got a perfect diffraction of the cactus to hand. So our wide field fluorescence microscopy image is one very complex image, right? It's got all of our fluorescent molecules emitting their fluorescence at the same time. One image, complicated, and our resolution is limited by diffraction. So in single molecule localization microscopy, we set up our labeling so that we have fluorophores that can go between an on state and an off state, emitting and dark. And so in the image acquisition, your first frame might look like this, only a very small proportion of your fluorophores emitting, they're quite well separated. In your second frame, a different random subset of molecules emitting. And you end up with this big old stack of data, these thousands of frames where you're just randomly turning your molecules on and off. What's the point of this? Great question. Well, if we can see single molecules, then we can very accurately estimate where the center of those molecules is, okay? Much better than we can in our original very complicated image. So we do something called detection localization. So each frame, we convert from this kind of image of blurry point spread functions into tiny little dots that we're very confident where the molecules are. Do that for each frame, and you end up building up this image of your super resolution structure. And that's what it would look like from all of these frames. Okay, so clearly there's a big bioimage analysis component here. We've got the whole thing hinges on computation. It hinges on us being able to accurately detect and localize these molecules. So it's a two-step process. Detection, finding approximately, it's basic peak detection, right? Which blobs look like they're the right size and shape to be a single molecule. And localization, which is then refining that very precisely to where the molecule is. And again, in this kind of period of boom for single molecule localization microscopy, kind of in the 2010s, anyone who is anyone in the field developed their own method to do this, right? There was this huge explosion of techniques for doing these steps, detection and localization. So much so that there was not one but two benchmarking papers on the performance of these methods. And the data I'm going to show you here is just a tiny subset of the data from this paper called Super Resolution Fight Club, great name. And this is kind of what one of these looks like. So I did it for lots of 2D and 3D data, lots of different data sets. This is just for one 2D data set, just to make things clear. It's actually for the data set up there. And each cross is a different algorithm, okay? Different way of doing exactly the same task. On our x-axis, we have essentially how accurately you're getting the localization, where smaller is better, the error in your lateral localization. And on our y-axis, I've plotted the detection efficiency, so the Jacquard index from this simulated data set of how accurately can you find each one. So you want high Jacquard and a small root mean square error. And you can see, firstly, there's a lot of these methods, right? And this one, there's 40 different methods alone. And this is from really nice. They've got a lovely web tool for exploring this data set. Okay, so this is great, right? If we're talking about precision and accuracy and errors, what a fantastic tool to help us navigate this field, right? We know exactly which methods we should be using, and they're clearly the ones that are right up here, right? Okay, so let's explore this data a bit more. So what have I actually come across in the decade or so I've been in this field? I'm really caring about the analysis side of this field. So those ones are ones that I've kind of intended to use, but never got around to using. I've always been like, oh, I should try them out. These ones, tried to use, but couldn't get the software to work. And one of these I use for my actual data, and it isn't even the best one, okay? And this is coming from someone who should know better. You know, I should really care about using the best performing algorithm for this, but I use the algorithm that's just in Fiji. And I think there's a little moral here, which is, again, people like me love doing this kind of thing. We love benchmarking the crap out of algorithms and data sets and saying what we should use. What people will actually use is the one that's easiest to use, okay? We will use what is convenient, not what is accurate. Is this a problem in single molecule localization microscopy? I'd say probably not so much. This is from a simulated data set. You can see there's a bit of spread in the error. That spread in that error is vastly outweighed by all of the things you do in your experiment, okay? The error in your data will be so much bigger than the error in your localization precision almost all of the time for a realistic data set. So that's just a little kind of moral to start with of how much should we care about error and localization? If things are benchmarked, do people actually listen to that? Okay, so I said that how we set up our experiment actually contributes a lot of this error. So if we go back to this, one thing that in microscopy we're always trying to navigate is this kind of pyramid of frustration, which is where you try and get close to one of these corners and you move further away from all the other ones. And you always have to compromise when you're doing the experiment, especially in a technique like single molecule localization. So we've seen here that the big win in these methods is the high spatial resolution, about 50 nanometers, which is very routine. But taking all those frames takes a long time, right? Minutes to hours sometimes. Again, this is just a random data set. So your sample probably isn't going to be alive or moving. And again, the conditions that you need to actually make this blinking happen are very phototoxic. Cells do not like this, okay? There's a lot of laser and there's a lot of buffering and there's a lot of sadness. So when I was in Ricardo's lab, we thought, okay, well, These methods are great. There's all these fantastic, accurate methods for finding localizations, but what if the data doesn't look as good? Or what if we want to acquire data under conditions where it's just not possible to get data like this? Okay, so what happens if we try and make it more live cell friendly, for example, with the data? So this is real data. This is Alexa 647. It's blinking on some fixed microtubules. And that's a classic single molecule data set, very high laser intensity, and you can clearly see the single molecules. If we want to make it slightly more live cell friendly, you might just think, let's just turn the laser down a bit. So if you turn the laser down a bit, then you see this. You lose those single molecules. If you turn the laser down a lot to make it super live cell compatible, then you see this, right? You lose the single molecules, and you just see kind of noise and random fluctuations in your fluorescence. Now, what happens if I try and analyze these with those wonderful algorithms that we just saw? Well, the first data set, as you can see, you get lovely microtubules out, right? That's what we expect. But if I put these two data sets through that exact same algorithm, we see what can only be described as catastrophic failure. Because the algorithm can't see things that look like single molecules, it can't detect them. If it can't detect them, it can't localize them accurately, and you just lose loads of stuff from your image. So data is king here, right? If your data doesn't match your algorithm, you're in real trouble. So again, one of the things that I did in kind of my previous life, kind of postdoc, not kind of, yeah, I don't think I've been reincarnated yet, is trying to develop an algorithm that worked with these data sets. And this algorithm was called SURF, Super Resolution Radial Fluctuations. And the idea was that you didn't need special blinking fluorophores, you didn't need a special microscope. You could just go to your wide field or confocal fluorescence microscope, acquire a time series, and run some image processing, and it would convert you from some kind of sad-looking LED wide field data into this improved contrast, improved resolution data set. And we could also do this, if I could find my mouse, there we go, for live cells. So you can see you can image for quite a long time, and it's just a computational method. So this was great, and we went around saying, oh, this is, wow, we've saved microscopy with computation, with bioimage analysis. This is brilliant. We can get super resolution from worse data sets. Okay, great, well done us. But again, the question we should maybe ask ourselves is the thing that I've developed actually useful, right? Because you've got this magic button now that you can press, and you can visually improve your data. So is it useful? So this is a data set that we included in the original paper way back in 2016. And this is a T cell expressing LIPAC GFP to image the actin. And you can see the difference between a TURF movie and SURF processed movie. And this is a really light sensitive system, right? T cells don't love a lot of light. They will die and not do the thing you wanted them to. So, okay, looks better, but is there more information in the SURF processed image? So one thing we wanted to do was we actually wanted to see if there was a direction to how the actin was moving in this system when it was dropped onto an antibody-coated cover slip. And we used this image analysis technique called particle image velocimetry, which essentially tells you if there's a direction over time to where something is flowing. And you can see if I run this on these two data sets, you can see SURF has clearly increased the amount of information we can get from our data set, which is great. If we don't do the SURF processing, then it's really difficult for this method to pick up on the direction that our actin is moving. Whereas here, you can see this, we've kind of recapitulated this color map, which is telling us that actin is flowing towards the center of the cell. Fantastic, SURF's really useful, great, everyone should use it. Okay, so it can increase the biological information in your image. But I'm going to show you another image, and this is one that we used to use in presentations all the time, just as a demonstration of how the algorithm works. So here's some microtubules, here's our wide field, oh, blurry, sad, and here's our SURF image, wow, contrast, good. And great, you know, it looks visually like the resolution's better and you've got better contrast. But I would ask you, is there anything that you can measure from this SURF image that you can't measure from this wide field image, right? If you're looking at microtubules, maybe what you might want to do is trace the microtubules. All right, sure. There. Okay. I haven't really gained anything by doing this SURF processing here. And actually, when you're being critical, you lose stuff, right? What's this hole? It's a computational method. It's not perfect. It's got lots of artifacts. So this is a huge problem in bioimage analysis. We've got all these tools, but when do we actually need to use them? When can they actually help us? And when might they just generate something that looks nice, but has a secret error in there? And again, coming back to it, obviously there's these problems, these things, but loads of people use Surf because, spoiler alert, it's a Fiji plugin, right? And that's kind of the curse of open source software development, is if you make your method easy to use and open source and free to use and very accessible, people will use it, which is great. But the problem is people will use it wrong as well. If there's a very low barrier to entry for anything, people won't read the manual. They'll be like, oh, a magic button, I've pressed the magic button. So this is where I guess bioimage analysts come in, is you are the bridge between the magic button and things making sense. And I spent a lot of time over the last few years fielding emails from people who have sent me data in their surf reconstruction saying, is this good? I'm like, what are you trying to achieve here? So again, just something as easy and accessible to use doesn't mean it's accurate and or useful. Okay, so obviously to help develop Surf, and it does have loads of good uses, but it can be used wrong. And so the obvious thing when you develop a method like this to do is to then have a huge crisis of confidence over the method and think, okay, well, how do we know if a super resolution image is good or not? So one way you can go down about this is kind of the thing that you saw in the challenge data, which is a simulation, right? You can simulate some data set, you can make it blink magically like I did with the cactus, and you can benchmark different algorithms on it so you can see how well an algorithm performs. Great. But what if you don't have a simulation? Well, there are also biological structures that we know very well that you can benchmark these methods on. So for example, the structure of the nuclear pore complex, nice and 3D, nice and regular, all the geometries are very well known. And there was a beautiful paper from 2019 from Jonas Ries's group where they actually benchmarked loads of different super resolution methods on how well they could image and recapitulate the structure of nuclear pore complex. Okay, so we can tell if our super resolution image is good. We have these systems. But huge problem here, what if you don't know what it should look like, which is the whole point of doing microscopy, right? We don't do microscopy to get something we already know. Okay, so how might we tell if our super resolution isn't good? Well, it's super resolution, and so we're interested in resolution, right? That's the thing, the reason we're doing this. So how do you measure resolution? Method one is you get your images, you find the bit of the image that looks best, and you draw a little line across it. And you pick your wonderful little bit of your image, draw a line across, and oh, brilliant, fantastic, 75 nanometers, nailed it. Obviously, this is not ideal, right? This is so subjective. You could easily draw it across a different part of the image and get a totally different result. So obviously, boo, don't do that. Right, is there a more objective way of measuring the resolution in these data sets? Well, there's a method that's been stolen from electron microscopy called Fourier ring correlation. So this is just from a paper describing the method. And here's a super resolution reconstruction of some X structure. And what we can do with this method is, this is a rendering, right? Our single molecule localization data isn't actually an image, it's a list of coordinates that we turn into an image. So what we can do is, instead of plotting all those coordinates in one image, we can just randomly split the coordinates into two and plot two separate images, okay? What we can then do is look in Fourier space and essentially see which frequencies match between these two images. So the highest frequency at which these two images match should be the resolution of your image. Anything beyond that is noise. So this Fourier ring correlation goes out, it draws rings in Fourier space at different frequencies, and it sees at that frequency, how well do my two images agree? If I go out a bit more, how well do the two images agree? And you make a little plot, and at some point it drops off, and the point at which it drops off is the resolution of your image. Fantastic. And this is used all the time to say, what's the resolution of my single molecule localization data set? Okay, so that seems way better than method one, right? Because we can just treat all our data at the same time. Okay, well, here's a little bit of simulated data. It's a little V-shaped structure. Super resolution reconstruction of this does not look good. You can see it's turned into a kind of wishbone structure. It's actually this kind of little hairpin V shape. All right, let's do Fourier ring correlation on that. Right, I split them out. And there's a problem here. And that problem is that it's wrong on the same spatial scale in both images, okay? It's this bit here, which is wrong. It correlates very well with that wrongness. So your resolution will come out as, oh, great, 46 nanometers resolution. Whereas if we went to a dumb method of 1, we'd say, well, actually, about here is where the closest place we can measure that separation, and that's 180 nanometers. So there is no good way to measure resolution in single molecule localization data, I'm afraid. Does that mean it's not worth doing? No, but I think trying to be measured in how you measure resolution and appreciating the pitfalls of these methods is super important and really important for users as well, because they will want to say, what is the resolution of my image? So resolution measurement is not ideal. Are there any other ways you can check how good your image is? Well, this is normally how you do it. You put your image next to a wide field image of the same structure, and you go, great, looks the same, probably good. So another method that I developed back in my postdoc was this method called SQUIRREL, which is an acronym for Super Resolution Quantitative Image Rating and Reporting of Error Locations. Yeah, well done, me. That's a huge amount of regret. So what SQUIRREL does is it essentially says, right, you've probably got these two images. If our single molecular localization image is perfect, if this is artifact-free, if this perfectly represents my structure, if I pass it through a virtual microscope, so scale the intensities, blur it out, then we'll get what the wide field would look like if it was perfect, which is this. And then, of course, we can just correlate the two. So our real wide field image and our theoretical wide field image, and differences between these will tell us this error map, so highlights where there are discrepancies, and some kind of magic metric which will tell you how similar are. This is based on the Pearson's correlation coefficient. 0.929, wow, must be good. Okay, so that's one way we can try and assess the quality of images. Get a map, and people tend to use this in the literature for basically saying that their method is performing better than someone else's method. That is the main way SQUIRREL is used. And I know that's not really how I wanted it to be used, but well. Okay, so what is maybe a useful way of doing this kind of error assessment? So again, as bioimage analysts, we often end up on the data side, right? We don't often, we might get involved in the acquisition, but sometimes you just get a load of data given to you. Okay, so these are neuronal actin rings imaged with DStorm, and this is what they look like in wide field, just kind of mush. And here's some raw data. And now a question that a microscopist or a biologist might have is, well, how many frames do I need to image in order to get a nice super resolution reconstruction? If you go for 500, that's clearly not enough. You haven't sampled your data fully. You're just missing loads of stuff. If you go to 60,000, then, oh, brilliant. We start to see these stripes come out. Actin forms these rings along the neurons. But is 60,000 enough? Is it too much somehow? Is it perfect? So one thing you can do with SQUIRREL is actually see when your information stops getting better in your data set. So what I'm showing you here is just the image, and I'm adding 500 more frames of localizations each time. Here I'm plotting the SQUIRREL quality, higher is better, and here I'm plotting the FRC resolution, which we know is flawed, but it's the only real way we have of measuring it. So as you add more localizations, our quality improves, our resolution improves, but then we plateau, right? We're adding more data, we're not getting better. And eventually at some point, something weird happens, right? We get better, we taper off, and then our quality decreases, okay? So we always think more data is good. Sometimes, especially in these kind of techniques, more data can kind of be bad. In fact, this was kind of where the quality peaked. It's only just half of the data set was kind of useful. And just to kind of show visually the difference between reconstructing from where the quality peaked and the whole data set, there's not a visual difference. So here, SQUIRREL is kind of useful. It tells us that over half our data isn't actually useful. It's actually making our quality worse, which is not what we wanted to happen. Just as another sanity check, we can do some other analysis just to see if this quality metric tallies with the information in the image, so how well you can resolve the difference between the two rings. And the information in the image peaks at the same point as when our SQUIRREL accuracy peaks. So this feels like SQUIRREL's useful, right? This feels like we should be able to, if a user comes to us with single molecular localization data, we can run this algorithm. We can see, oh, okay, is it good? How much data should you have used to reconstruct? Fantastic. But is it useful? So again, once again, I find myself asking, is the thing I've developed actually useful? So here are some microtubules. This is real data, and it's from one of the super resolution challenge data sets. And here they are blinking. And it's quite high density blinking, so quite a few of the molecules are overlapping, which is not ideal. So if I reconstruct this data with thunderstorm, which is that one green circled cross on that plot, because again, it's very easy to use, I get a reconstruction, fine. I can get my squirrel error map that says, it's kind of okay, there's a bit of dodginess happening up there. Another thing we put into Squirrel was local FRC, so you can map the resolution across an image. Okay, great, well, we can see that something weird is also happening in the resolution around this place, so that bit's probably quite bad. All right, cool. We kind of trust all that output. Okay, great. So a few years ago, there was this method that came out called Hawk from Susan Cox's group. And it's just a pre-processing step, single molecule localization data that tries to spread out in time a bit more using wavelet filtering. Okay, so what does the Hawk reconstruction look like? It looks better, right? We're losing less stuff. But Squirrel says it's worse. Okay? It's worse because Squirrel is very good at telling you where things are linear with respect to your diffraction limited data, but not necessarily some of the structural things, which is not great, right? It's saying there's error here where it's quite good, but that's because we were just brighter in our original image, and that hasn't been picked up by the super resolution data. And again, our FRC map is kind of telling us a fairly similar story, right? So this is to say that Squirrel is kind of useful, but take it with a pinch of salt, all right? It seems to work for quite a lot of things, but sometimes, depending on the algorithm you use to reconstruct the data, sometimes it just doesn't work that well. Susan's lab also developed this method called Hawkman, which tries to actually look at how well things match or between unhawk processed and processed data on different length scales. So how confident are you on different length scales? And this is a technique called Hawkman. So here we're increasing the length scale on which we're looking. So at 20 nanometers at resolution, nothing really agrees. And it gets more confident of how similar basically these two images are. Around maybe 80 to 100 nanometers. So again, we're starting to get this kind of nightmare situation where we've got all these different quality metrics piling up, okay? I've given you Squirrel, which we've seen can be useful, but also can't be useful. We've got FRC, which can be useful, but also can't be useful. And we've got Hawk, which can be useful, but you need to do the Hawk pre-processing. It won't work for any other super resolution technique. So what we're trying to do at the moment is Susan and I actually have a joint grant to try and get this all into one package, right? So a kind of one-stop shop for single molecule localization assessment and making it really clear where each of these different quality assessment techniques, what the information tells you, okay? Because it tells you different information. These three different techniques tell you different information. And all these techniques are, of course, available in Fiji. Okay, so what if the reference image is crap is a question that I started thinking about during the pandemic because I had nothing else to think about. And I thought about it so much that I wrote an entire grant to make a research group to answer this question of what if the reference image is crap. So this is what we expect in Squirrel. We expect to have a super resolution image where we don't really know what the quality is and a reference image that we trust, right? We trust diffraction limited data. We haven't needed to do any weird pre-processing. And so here, it's a bad match and Squirrel will tell you it's a bad match. Correct, things all agree. If we've got a better image, then Squirrel will hopefully tell you it's a good match. Okay, that's what we want. But what if our reference image looks like that? Okay. Squirrel is not telling you anything about if your super resolution image is good. It's just telling you how well it matches to your reference image. So if your reference image is bad and your super resolution is bad, everything will say it's bad, but maybe not for the right reasons. And if your super resolution is actually quite good, but the reference is bad, Squirrel will tell you it's bad when it's actually good, okay? So I spent a lot of time thinking, well, how can we legislate for this? How can we legislate for the fact that someone might put in a slightly dodgy reference image?